Chad.
Hey, Dan, what's up, man?
That's hilarious. We're the only ones here.
I know.
Oh man.
I always hop on early.
Yeah, I gotta do the intro.
Oh, yeah?
Yeah.
I have back-to-back meetings today, like this one, then one, then two, and then have to be Gardendale at 3. So super busy.
Yeah.
Man, it's good to see you. How's work been going? Y'all staying busy?
Yeah, I was on it's kind of hit or miss for us. I mean, it's always busy at the coroner's office, but we we have certain days where we're on service and off service. And so if we're off service, we generally try to protect that time for each other. So I was on service for 6 straight days, which is more than usual, and I just got off service. And so the rest of my March is actually pretty light. So it's kind of hit or miss for us.
That's good. I was the same scenario. I finished up my last or 8th straight day of call yesterday. Last night, actually.
Oh, really?
Yeah.
Just a busy time for you, I guess.
Yeah, super busy. And we have new residents too, so that's always busier time.
Yeah.
They don't really know like what's going on.
I know. Yeah, you probably trying to get my, let's see here, turn that down. Yeah, the residents that come through here, they're here for like a month, and it's probably different than you having to train a resident, and then I'm trying to be careful what I say in case anybody else hops on. I guess this is recording, but it's different from training. Like training them for just for a month's purposes is different than the whatever 3 or 4 months I'm sure you do. So
yeah,
the scope is different. So this is recording. Maybe we should like hop off.
Yeah, you can stop the recording, right?
Well, pause. And probably what we'll do, usually people will type in questions throughout the lecture. And then, so if we have a few minutes for questions at the end, I'll probably go through those questions first and kind of direct us through those. And then after that, we can do some live questions, obviously.
Okay, I tried advancing my screen and I couldn't do it. Let me try this again.
Okay. So right now I just see your sort of your PowerPoint. Not in presentation mode. There you go.
Okay, there we go.
Yeah.
It's working.
Okay, it is noon, and so we're going to go ahead and start our weekly grand rounds. Joining us today is Dr. Stephen Austed. He currently serves as the Protective Life Endowed Chair in Healthy Aging Research and is also the chair of the Department of Biology here at UAB. He is the Senior Scientific Director of the American Federation for Aging Research, which is a New York-based national philanthropic organization. He received his Ph.D. from Purdue University in 1981. He accepted his first faculty position as an assistant professor at Harvard University in 1986. He then moved to the University of Idaho and became full professor in 1997. In 2004, he was recruited to the University of Texas Health Science Center in San Antonio. He became director of the Barshop Institute for Aging and Longevity before coming to UAB in 2014. He's published over 200 scientific papers and book chapters. His research encompasses many aspects of the biology of aging, from the molecular level all the way to the epidemiological level. He's well known for his work on identification and study of exceptionally long-living non-human species for insight into the science of slow aging. He's won many, many awards, including the Nathan A. Schock Award, the Robert W. Kleemeyer Award. He's also served on the Scientific Advisory Board of NPR, written some 150 essays for the general public on the science of aging. He has a couple books, one, Why We Age, which has been translated into 9 different languages. He has got a book coming out this year called Methuselah's Zoo, What Nature Can Teach Us About Living Longer, Healthier Lives. Needless to say, Dr. Austed is well-rounded with an undergraduate degree in English literature and also has experience in construction, journalism, billiards, taxi driving, and line training for the film industry. And so he's going to present sex differences in aging and resilience. We're really happy to have you here, Dr. Austed.
Well, thank you, Daniel. Thanks for the nice introduction, and welcome everybody. I got interested in sex differences quite accidentally a number of years ago, and I'll explain that to you as we get started. First, I'd like to give a little background on aging and basically explain why everybody should be doing aging research, not just me. And I know in medical school you learn that aging is the biggest risk factor for most chronic diseases, but I don't think we ever really get a visceral idea of how big it is. So let me just start off by trying to give you some visceral feel for that. So here are a number of risk factors for death by heart disease, and they're the typical things that the NIH is trying to get us to get under control. Let me just put these in context by adding in aging to those. There you go. They're all the now aging in this case is basically a dying after 65 as opposed to before 60. And you can see that all these risk factors really become trivial when it comes to aging. Now I could do this for disease after disease after the disease, but just let me do one more. And this is Alzheimer's disease. And don't worry, having sex is not a risk factor. The risk factor there is, depending on what sex you are, there's an increased risk of developing Alzheimer's disease if you're female. There's also diabetes, physical activity, and the ApoE4 allele, which is shown here, which increases the risk of Alzheimer's disease by about 5-fold and cardiovascular disease substantially too. Let's put all of these diseases again in a perspective, and as you can see, they all disappear into the x-axis, except for ApoE, which you can still basically see. So I think that gives us a background for really why aging should be thought of as the #1 global menace to health, because it underlies basically all of the things, most all of the things that kill us and all of the things that degrade the quality of life. As we advance into the 21st century and continue to becoming longer and longer lives, we're already by far the longest lifetime in history. I think we can see the later life misery indicators increasing, not just death. And if you look at cataract removal or these various replacements, you can see that these are things that people have because they're causing them a great deal of misery. And wouldn't it be nice if we could somehow delay or prevent some or all of those? And that's what really trying to understand the basic biology of aging is all about. So I think that the 21st century brings with it an incredible aging research imperative, because if we don't understand something about aging and how to modify its effects, and we only attack disease by disease, we're looking at a great deal more dementia, frailty, osteoporosis, all these things that again degrade the quality of life in our later years. And also, let's not forget that the kind of massive healthcare costs that this will enable. So that's setting the outline of why aging is important and why we have this new approach, which is targeting aging rather than addressing specific diseases. And I'm not going to talk about that as much as I'm going to talk about some surprising findings that have emerged over the last few years, which suggest that there are more differences between the sexes in aging and resilience than I think we ever suspected in the past. So let me start with the sex differences in resilience by talking about the Donner Party disaster of the winter of 1846 and 1847. If you grew up in California like I did, this was in all of the history books. And this was a group of pioneers that in the middle of the 19th century set off from the Midwest for California, hoping to make a new life for themselves. They had no particular survival skills. They were poorly led. They were poorly equipped. And they ended up getting stranded by early season snows in the Sierra Mountains in February of in November of 1846. Like I said, it was a lot of early snows. And they'd made several attempts to try to get out of there. All of these were unsuccessful. And by the time that they were finally rescued in early 1847, about half of all of them had died. Most of them had died of starvation and associated diseases. Some of them had actually been murdered. But the striking thing to me, when I went back and I reevaluated that, is the difference in the fraction that survived this catastrophe. As you can see from the data at the bottom here, fewer than half of the men survived, but more than 2/3 of the women survived. Now you can imagine a number of reasons that women may have been better survivals during this particular ordeal. They may have been more resistant to starvation. They may have been preferentially treated by the men, and there may have been some vestige of chivalry that remained. Or the men could have simply been more violent with one another. But the key thing about them is that they ended up eating the deceased, and by the time they arrived, they became quite infamous because of the cannibalism that they've done. So like I say, one can imagine some reasons to see this sex difference in resilience in this case. But a few years ago, there was an analysis of resilience under a far broader range of conditions. And this particular paper here showed how there was biased female survival in some of these massive catastrophes. Some of them were famine, and some of them were epidemics. And I'd like to just point out a couple in particular. In the Irish potato famine that lasted about 4 or 5 years, that killed about 1 million people and caused the immigration of at least that many, life expectancy for men during that famine fell to 18 years. And to women, of course, it fell substantially as well, but it did not fall as far as for men. Similarly, in the Ukrainian famine of 1933, 1934, which is really caused by Joseph Stalin, the Russians have not been torturing Ukrainians only recently. In that particular famine that was caused deliberately by Stalin, about 4 million people died. So many people that men's life expectancy fell to only 7 years. But again, women survived better and survived 11 years. And this wasn't only the cases for famines, it was epidemics. And I think we're all aware the fact that COVID-19 has taken a slightly higher toll in men than it has in women. Not accidentally, I'm showing you pictures here of the longest lived 10 humans that we know of. And one striking feature about them is that they are all women. By the way, this woman in the middle on the bottom, Sarah Knaus, is the second oldest woman, 119 years and change. This is Jean Calment, 122 and a half years. And this is Take Kanaka, who sometime next month, if she survives that long, will pass Sarah to become the second longest live person. When one gets over the age of 115, the survivorship is 95% female. So let's just look at exactly how robust this is through the women's survival advantage in one particular country. This is a country that I only chose because it has a circumscribed, easily monitored population, and they've been keeping excellent data for many years, and that's the country of Iceland. Iceland, looking at the top panel here, in some of the years where it suffered from epidemics, life expectancy at birth has fallen to as little as 18 years. And in the 1980s and early 1990s, Iceland was the longest lived country in the world, subsequently replaced by Japan. But one thing to notice about it is that in each and every year over that time, women survived better than men, and they survived better if you considered their survival from birth or if you considered their survival from age 50. Parenthetically, one thing I analyzed as I was looking through this data is I thought, what about survival from birth to age 5? And it turns out that women universally survive better. I guess they wouldn't be women at 0 to 5, they would be girls or babies survive better. And in fact, in prematurely born babies, being a male is considered a risk factor for death. So I hope I've convinced you now that this female survival advantage is there through life. It sometimes gets bigger, it sometimes gets smaller. In the case of Russia, it's been the difference, and it's been over a decade. In the smaller countries, it's 4 or 5 years. It's currently about 5.5 years. And this is really one of the most robust features of human biology that we know of. And for some reason, everybody knows about it, but nobody's thought about it that much until relatively recently. The sex longevity gap may narrow. One keeps hearing it's narrowing, and in the US, that's true. But in Japan, for instance, the gap is actually growing. So I see there being no prospect of it changing anytime soon. Now, one of the major questions, of course, is why is this the case? There are some obvious answers. Men differ in some ways. There's this period of testosterone dementia that we undergo in our teens and 20s, and some people into our 30s. But one of the interesting things about it is that it's not really due to 1 or 2 causes of death, 1 or 2 pathologies that men are just prone to. And what I'm showing you here is for data from 1916, actually the data would not look any different today, with the exception that COVID would be in there. And anything above that dotted line means that men are dying at a higher age-adjusted rate. So this is not due to the fact that women live longer. The only exceptions are stroke, and that's not really an exception. It's just that the sex difference is very, very small, but men die at a slightly higher rate of stroke. But there's this striking exception of Alzheimer's disease. And we don't really understand exactly what could be causing that. There are some hypotheses. There was a paper out in Nature just this week, or last week, I guess by now, attributing it to elevated FSH, postmenopausal elevated FSH. And that was a mouse study. And I hope I convince you before my talk is over that mice are not the best measured species, I think, for understanding sex differences. But we'll get to that in a second. I also want to point out some other ways that sex differences are driving changes with aging that you may not be aware of. It turns out that in virtually every neurodegenerative disease, there is a sex difference. Quite often, it's in the direction of males, as in ALS and Parkinson's disease, but occasionally in Alzheimer's disease, and certainly in autoimmune diseases, women tend to be struck more often by autoimmune diseases like MS. But even in things that we don't think about, here's a study from UAB, actually, looking at the decline in muscle fiber type with age in men and women. And I sort of highlighted the general trends with these bars. And you can see for each of these 3 survivor types, there's a difference in the pattern between men, which is the blue lines, and women, which is the red lines. So for instance, for type 1 type muscle, there doesn't seem to be much of a change with age in women, but in men, the fiber diameter actually increases. And as you can see for type 2X, there's no change later in life in men, and there's a decrease in women. Again, this is just one of the many bizarre things that emerge when one starts to look at both sexes throughout the life course. And I'll leave you with one other one, which has to do with pain. It turns out that self-reported pain is virtually dominated, whether it's back, migraine, musculoskeletal, neuropathic, whatever, by higher reports among women, anything above the line. And these 20-some studies means it's higher in women, which is not particularly surprising. There can be lots of sociocultural reasons for these sorts of things. What is surprising is that for some types of pain, there's actually a mechanistic difference in the way that pain is experienced and in the way that it can actually be treated. And I give you this slide here, which comes from the work of Rob Sorge, who is here at UAB in the Department of Psychology. He did this before he came, but basically what he did was look at neuropathic pain in mice and noticed that drugs that treated neuropathic pain by inhibiting microglia were effective in males, but not in females. Not knowing what to attribute that to, they actually castrated some males. And when they castrated the males actually failed to get the same kind of pain relief that the females did before. They failed to get the same as the males got. So this is a fundamental mechanism of pain perception that is actually sexually dimorphic. And what it means is that there will be certain analgesic agents that work well in one sex and don't work in the other. And I was shocked and surprised to learn this. So of course that brings up the point, since 90% of all of our biomedical research, certainly almost all of it is invasive, is used as laboratory mice. A question that immediately comes to mind is, can mice be informative about these sex differences? Are they the right model? Are they a model we can trust? And so I'll ask you a question. I don't know how many of the people in the audience work with mice, but I'd like to ask the question, do male or female mice live longer? Are they like humans? Do female mice live longer? Is there no difference, or do males live longer? This is actually a question that occurred to me when I was reviewing the literature about a decade ago, and I found two papers that came out in the same year, one of which said the conceptual basis of this paper is trying to understand the consequences of male mice living longer than female mice. Second paper, using the same C57 black 6 mouse, said, we chose this strain because everyone knows there's no difference in the longevity between the sexes. So it turns out that caused me to go and dig into the literature and dig up about 120 studies of mice, looking to see, do male mice or female mice live longer? And it turns out the answer is yes. Male mice and female mice live longer. So this is about 90 studies, and as you can see, they range all the way from males living 40% longer than females to females living 20% longer than males. Now, if we understood this range, it might be that we could learn a great deal. If we understood what determined, the immediate assumption that I made was that this was due to genetic background. And if we actually control for genetic background, we might see a much clearer pattern. However, it turns out that if you look at the most commonly used background, the C57 black 6 mouse, you find exactly the same type of result. Not quite as extreme, but one finds things all the way from males live longer to females live longer. And I went through and I did a number of other backgrounds, any that had a variety of studies, and found exactly the same thing. Now, again, if we understood what the conditions were that favored males versus females, it could be that mice could be quite informative about understanding the mechanisms underlying these sex differences, but at least as of now, we have no idea. Once I started looking into this, things started emerging again in the biology of aging that surprised me. One of the things was that some of the genetic manipulations that we were doing that lengthened life and preserved health had effects in only one sex. I'll give you an example here. This was a mouse study where they knocked out a gene, S6K1, which is one of the effectors in the mTOR network. And by doing so, they found a robust effect in survival in females on the top there, and no effect in males. And it turned out in, again, females only, there were also health benefits, but again, no benefits seen in males. Now you might think, is this, I mean, this is weird. The same gene was knocked out in both sexes. Why is it having an effect only in sex? It turns out if instead of knocking out a gene, you lengthen life by, let's say, overexpressing a gene, you come out with something like this. This is a gene called SIRT6, one of the sirtuins that was overexpressed in several genotypes. I've shown you two of the overexpression lines here. And what is circled on the left there is the survival advantage that's seen in the male lines only. And on the right there, the survival lack of advantage that's seen in females only. And again, the transgene was expressed the same in both sexes. So what's going on here? This really tipped me off to the fact that there were some mysteries to understanding sex differences, particularly when it comes with aging, that we just had no clue about. And then there was the interventions testing program study. Let me explain what this is because you may not be familiar with it. This is an National Institute of Aging supported program that will test drugs that you think might be extending life in mice, and it will do it for free, and it will include you as a co-author, and it will do it simultaneously at 3 different sites. The idea being that by triplicating the experiments, one will get immediately verification if the result is true or not. And this is just 3 other sites in the University of Michigan, the Jackson Labs, and University of Texas, San Antonio. And so far, these, I actually haven't updated this in a couple of years, but the important thing to note is that, first of all, there's been quite a bit of success. About 1/3 of all the drugs tested have actually substantially increased longevity, or let me say significantly increased longevity in about 1/3 of the animals. But the more amazing thing, I think, is the fact that in most of these, it's affected one sex only. So for 17-alpha estradiol, there's been a 20% increase in male longevity, 0 in female longevity. Aspirin at the top there is crossed out because they actually did this 6 times. They did it once and found a positive effect, and then to follow up, they repeated that at the 3 sites and didn't find the effect the second time. But this is really quite remarkable. And in even some of the ones that don't show a sex bias, such as rapamycin, there was a difference. And acarbose, there was a difference. So here's the results with acarbose. As you can see, there is a female, a small female effect on survival, but a much larger male effect on survival. And this has subsequently been replicated at several doses. Here is the same result at a different dose. Now there's no effect whatsoever. At a different dose, again, there's no effect. So we really are seeing this in time after time after time. We're also finding the effect in something like glucose tolerance tests, where there's effect in males, but no effect in females. That effect, at least in this example with 17-alpha estradiol, can be eliminated by simply castrating the male. So at least in this case, it seems to be a function of male reproductive hormones. Probably the most interesting, And the drug that, at least in mice, has had the far most robust effect has been the drug rapamycin. And as you probably know, it's used in a number of contexts in human medicine. It's used to be embedded in cardiac stents. It's used in some chemotherapeutic combinations. And in mice, at least, it has an incredibly robust effect on many aspects of aging, from survival, which is shown here. And as you can see, there's an effect in both sexes, but the effect, at least at the higher doses, this is showing 3 different doses of rapamycin, the effect is bigger in females. And what makes this particularly interesting, this is just summarizing the effect. And here I'm looking at 4 doses. I'm looking at the 3 doses that you just saw. And this, again, this is administered in the food in the interventions testing program. The 4th one here is actually a separate study that was doing rapamycin by injection, but it was doing it at a level that seemed to be about 3 times higher than the highest dose that was being given in food. The interesting thing is on the sex specificity effects. As you can see, at the lowest dose, it had 5 times bigger effect on female survival than it did on male survival. And as one increases the dose, that difference gets smaller. It's almost gone at 42 parts per million. And actually, by the time you get to the injectable highest dose, females are actually males are actually living longer than females at this dose. So there's an incredible richness of underlying biology here that we don't understand at all, and I think deserves some further consideration. By the way, the effects we're seeing here were not simply a function of blood levels, the doses. You can see the blood level at the 4.7 parts per million, which is where there was the biggest sex difference, actually was the most similar. So again, really interesting biology. What can we do with it? So we've also done some work on grip strength, and again, it varies depending on the sex. This is a rapamycin study. Now, all of these things, believe it or not, there are some very crude and human data that one shouldn't take all that seriously, except for the fact that the results are so huge. There have been 2 human studies looking at men who were castrated for one reason or another. On the left, it had to do with men in the royal court in Korea. And on the right, it had to do with men that were in asylums for insanity, basically. The thing to note is the size of the effect. In both of these cases, the men that were castrated were living on the order of 15 to 20 years longer on average than the intact men. Again, this is very crude data. It's poorly controlled. One doesn't know what to make out of it, except for the fact that it's such a big effect. Now, in both of these, we're in hundreds of individuals. This is not a dozen individuals. Both of these is hundreds of individuals. So that might give one pause. Now, continuing on the effect of rapamycin, I just wanted to mention that rapamycin has been incredibly effective in mouse models of Alzheimer's disease. Here's one of at least a half dozen studies that have happened now of this mouse here, which is a triple transgenic. It's got 3 human transgenes in it to try to replicate as much of the pathology of Alzheimer's disease as possible. You can see the tremendous reduction with rapamycin in this is A-beta staining, as I recall. And that, it turns out, they were not looking at sexes. One of the things in the older literature is that quite often sex isn't even mentioned. But in this particular mouse strain, where they weren't looking at, if you actually looked at how long the animals survived if you let them live out their normal life, it turns out there was a big sex difference in this case as well. So in this triple transgenic, the females lived about a third longer than the males did. So again, I am just trying to emphasize that there are all of these mysterious observations pointing to the fact that as a variable, and when it comes to aging biology, sex is one of the most intriguing variables that remains largely unbalanced. Now, of course, NIH has subsequently said in 2014 that you have to balance sexes in both cell and animal studies. This had been done in clinical trials for some time before that, but it finally got through that there's some interesting biology that we may be missing if we don't include both sexes. And as a consequence of that, there's been a tremendous increase in the amount of studies that are actually looking at sex differences. And you can see this stops about 2012. And if I had continued this out, it would be dramatically greater even now. So this is a hot area of research. I'd like to just clarify what I think the mystery has got to be here. You know, the fundamental difference between male and female mammals, at least, is that females have two X chromosomes and males have an X and a Y chromosome. As you probably know, one of the X chromosomes is so-called inactivated randomly in each cell of the body to avoid the dosage compensation that would be necessary because the males only have one. It turns out that that's a gross generalization, that not the whole X chromosome is inactivated. And the part that's inactivated is very, very different depending on what species you're looking at. So to better understand sex differences between males and females, we need to really do a much better job of understanding X inactivation, which has been studied a lot, but also in understanding the Y chromosome. Understanding the Y chromosome is one of the few things at which has been studied far less than the sex chromosomes in females. And so let me just tell you why I think the Y chromosome deserves more respect than it gets. One of the reasons it doesn't get much respect is that it always has been assumed that the traits that are remaining on the Y chromosome are mainly related to reproduction and male secondary sexual characteristics. But it turns out the human Y chromosome has 568 genes. Now that includes genes and pseudogenes. 71 of those at least have protein coding potential. In fact, we know of 27 that are unique protein coding genes, and some of the others may be. It just hasn't been studied sufficiently yet. There's over 100 long non-coding or short non-coding RNAs, and we now know how important these things can be for gene regulation. And then there are a whole bunch of pseudogenes on the Y chromosome. Now, that's still a lot fewer genes that are on the X chromosome. But if these genes are expressed not just in the gonads or places one might expect secondary sexual characteristics to be important, they could be important. And this actually shows that some of these genes, and I've pointed at them here. So on the Y-axis here, you see a whole variety of human tissues. And on the X-axis, it's showing where these different Y chromosome genes are expressed. And the ones that I've pointed to down here, these 9 genes, as you notice, are expressed in virtually every tissue. My thought is that Y chromosome genes that are expressed in every tissue deserve some consideration if we're really going to understand sex differences in aging or other things. So let me here finish up by talking about how I think we might be able to use. I'm still not convinced that we're going to be able to use the mice for reasons that they're just not going to be informative. But if we are, let me show you some tools here. There are, because of the way we can genetically manipulate the mice, Art Arnold in the early 2000s really developed some mouse models that he thought would allow us to separate what he called activational effects of sex chromosomes from organizational effects. And activational effects are reversible due to gonadal hormones like castrating or gonadectomizing one sex or the other. But the other effects are organizational. That is, they probably are things that occurred during development. And once they've occurred during development, they can no longer be reversed. And I think the most interesting model that he produced out of this was what's called the 4-core genotypes model. And let me just explain to you what that is. These are mice that have had the sex-determining locus, the SRY gene. SRY gene is necessary to get the development of male gonads. Females do not normally have it, and males have it. It's on the Y chromosome. What has happened is that they have gotten a mutant where the SRY gene has been vanished from the the Y chromosome, and it's now on one of the autosomes. It's on chromosome 3. At the same time, and what that does is that allows one to develop mice that have XX females, your standard mice, but also XY females. That is, females that have a Y chromosome. It simply doesn't have the SRY gene. So gonadally, they're females, even though they're XY. And similarly, one can get XX males that have 2 X chromosomes, but because they have the SRY gene on an autosome, are gonadally males. And then we have the normal males. And if you gonadectomize those, you now have 8 sexes. You have all kinds of combinations of activational sex, organizational sex, and one can then begin to do quite a bit of interesting research. So here's some of the first research that came out of this group. This is by Dina Duval from UCSF. And what it basically showed was that there is an advantage in having an extra X, even though that extra X is largely inactivated. And what we're seeing on the left panel there are simply the males versus females. Now, under the conditions that she raises her mice, and these are in a C57 Black 6 background, it turns out that females live slightly longer than males. What we're showing in the middle is to have XX and XYs that have testosterone. And here you can see that there's an advantage of having an extra X, even if that comes with a great deal of testosterone from the male gonad. And on the right, we're seeing animals that have only ovaries. And you can see in that case, even though XY males have ovaries, having the extra X chromosome matters. So this is just the first bit of study that goes on. There's a lot of other stuff I'd like to mention. I didn't know how much time I was going to elect. Well, I could have added a few more things. Let me just mention a couple of things. I have some serious reservations about whether we're going to learn a great deal from the mouse ultimately about this issue, even though we have these wonderful tools. And I say this for 2 reasons. Remember, I said that understanding the dynamics of gene expression on the X chromosome and understanding X inactivation is critical. It turns out that mice and humans inactivate different parts of the X chromosome. They don't inactivate all the same genes on the X chromosome. This, to me, is problematic. The other thing has to do with what genes are on the Y chromosome and what genes are expressed on the Y chromosome. And it turns out that there are a handful of genes that are expressed on the Y chromosome that are found in both mice and humans, but there are also a number that are not. And they are things involved in cell cycle, a bunch of very fundamental processes. So I know that mouse research is going to continue on this, but one of the things that I always advocate is to broaden the bestiary. There need to be other species. Part of the slides that I deleted, because I didn't know that I would finish this early, are some other species that I think would be of considerable interest. We do have species like the laboratory opossum, which is a marsupial. The interesting thing about marsupials is they don't randomly inactivate one or the other X chromosome. They always inactivate the paternal chromosome. So looking at the two sexes there might tell us something. Also, there are a couple of species of common laboratory animals, Chinese hamsters, for one, that were used for many years, in which males consistently live longer than females. And those would make a nice contrast to things like the laboratory rat, in which females robustly live longer than males. So I do think there are some novel directions to go. And then I think with all the modern tools that we use in human biology, there are some interesting ways to address human sex differences that have yet to be exploited. And so I'll finish up there, leaving plenty of time for questions. This is some of the people that have that I've worked on these projects with, and some of the people that have supported my research over the years. So I'm happy to stop there and take any questions that you might have. Thank you.
Thank you very much, Dr. Austed, for that very excellent presentation. Okay, a couple things coming in the chat right now. Jarish Milkani says, excellent information, and is wondering how much behaviors and environmental conditions responsible for sex difference.
Well, it's interesting. In the case of humans, that doesn't seem to matter. You find these sorts of sex differences basically in every country and every culture. And the only time you don't see them is if there's some serious effort to prevent women from ever seeing doctors. In that case, there is a difference, but it's a very, very slight one. In the case of the mice, there probably are some real subtle details of husbandry that are explaining that great diversity. I can't imagine what else it would be, since we have complete genetic control of the mice. So there's got to be some subtle aspects of that, and I don't know what those are. I've been meaning to actually put an undergraduate on a project to go back through those 120 studies and see if there's anything that we can correlate with that. The trouble is, a lot of those studies were done fairly early in rodent research, where the husbandry is not very well described. You know, modern husbandry, we work very, very hard in aging biology to keep any infectious diseases out of our colonies. In the past, they didn't do that. And some of this may have to do with diseases that people didn't realize had crept into their colony.
So can I ask you a question? So, very good talk. Thank you. Does estrogen has a role in sex difference in longevity?
Yeah. No, it could be. And I mean, I think estrogen and testosterone are the first two things that people thought about. And particularly since there's a lot of neuroscience that suggests that estrogen has neuroprotective effects, right? But here we are with women having, more Alzheimer's disease than men. What's that about? I have no idea. This recent paper that looked at that and came up with the idea that it may be the increase in FSH rather than the estrogen going away after menopause. That's a kind of intriguing thing to me. But yes, it could be that there's something about estrogen that's protective as a whole. There's one thing that I didn't mention because, limited time and space, but there's something called the morbidity mortality paradox in between the sexes in humans. And that is, even though women under virtually every circumstance that we know about live longer, they also are less healthy later in life. That is, they have they report more diseases, more pain, see doctors more often, take more medications. All these things that one might attribute to sociocultural factors, except for the fact that they're also very robust. They're seen in cultures throughout the world. So what's that about? One explanation, and I kind of like this explanation, is that women survive things that kill males. And so the women that are surviving the longest, that are after most of the males have died, it's a male survival effect. the males that manage to survive are males that are particularly healthy. And
So I think that's probably explain, I think estrogen effect because pre-menopause, estrogen levels high, but
right.
post-menopause, estrogen levels decline, and that's why cause a lot of health issues, right?
Right.
Yeah,
On the other hand, all mammals have estrogen, but females aren't longer lived in all mammals. So is there something that's unique about human estrogen?
yeah.
Right.
I
One of
think
the things one of the things I was going to say about this mortality morbidity paradox, a friend of mine has a study of centenarians, people that live to be at least 100-year-old. But to be in his study, you have to be healthy. You have to be living independently at least to the age of 95. Now, in most centenarian studies, there are 3 or 4 females per every male, just because that's the people that live to be 100-year-old. In his study, because you have to be a healthy centenarian, he has a 50-50 sex ratio. So that's also quite interesting.
Thank you.
Steve, thank you for that presentation. I had a question if you can comment on all the magical anti-aging supplements, therapies, drugs, and whatnot that are out there. Because, at least from the free radical side of things, there's a lot of, nonsense that's been proposed and propagated. And some really, high impact sort of scientists with a lot of name recognition have made quite a living on the side of this. But I was curious as to what folks in the aging community think of some of those efforts and whether there's any merit to it.
It is the bane of our lives, Rakesh. It is something that we deal with all of the time. And the problem, you put your finger on exactly the right problem. There are some presumably reputable scientists that seem to have gone over to the dark side. And I won't mention any of them by name, particularly because probably the best known of these has done a very nice endorsement of my forthcoming book that's actually on the front cover of the book. But this is a terrible development. And the reason I think it's so terrible is that now it's not just the crazy people like Dr. Oz that are doing stuff like that. It's legitimate scientists, and it's making, it's making the field a lot more difficult to separate the nonsense from the real science. It's hard enough for we scientists to do that, right? But now trying to convince the public, I spent almost an entire lecture in the aging course I'm teaching now on resveratrol, something that's the darling of the anti-aging business, something has virtually no evidence to support its impact, even in mice, much less in humans.
That's the one I was thinking about. So.
Yeah, I figured it probably was, and I know the person you were probably thinking about too. Like I said, I'm hesitant to name him since he did such a nice endorsement of my book, though. But so I won't name David Sinclair. I'm sorry, I just refuse to.
Garish, did you have a question?
Yes,
Or
so
follow-up?
I just wanted to follow up on that. I was specifically asking about smoking, one of the big factor in men versus women. Is there any study on that? And on top of that, like working condition, night shift in general, like I don't know how long we have that data. So more will be more on those day and night shift, like 50 year back, all that one. So do you know anything about that? And there is any study available for that, like that?
Yeah, we do actually, and we probably don't know as much as we can. And there was clearly a sort of socio-cultural overlay on the underlying biology, and that's probably why the differences between the sexes are much bigger in some places than other places. A lot of that probably has to do with histories of drinking and smoking. So, like I said, the greatest sex difference in longevity in the world is in Russia, and that's largely because of the short life expectancy of the men rather than the long life expectancy of the women. that probably has to do with smoking and drinking. Some of the sex differences in Europe, which has been relatively small compared to the US, probably, but those are likely, I mean, they've been relatively large to the US, but they're likely to be shrinking because women in Europe started smoking later historically than women. And they've also been slower than the US, they've also been slower to quit. So that means that they are probably facing some demographic consequences in the next several decades that are going to change the amount of sex difference, but it's not going to go, it never goes away no matter what.
Yeah, the question is like more on that, the condition also, because I work on circadian rhythm, how that influence aging, sleeping, waking pattern, eating pattern, all those affect, aging significantly. Now, if there are any difference, any data available that can be used in animal model. So that's what I want to use, to prove what is the mechanism, how that.
Yeah, in terms of shift working and things like that, I don't think there's been sufficient evaluation of sex differences in that yet. I'm sure there's probably an impact. I don't think that it certainly is not a sufficient impact to make the difference go away, but it could affect how broad or how narrow it gets. The thing that I say that strikes me most is that in every country, in the countries where life expectancy is 50, and in countries where it's 84, you see exactly the same differences, or you see the difference. It's not always the same amount, but you see the same difference.
Thank you.
Hello, can you hear me?
Yes.
Yes.
I can.
This is Bill Grizzle.
Hey.
I give a comment and a question. The comment is, scientists, even in this meeting, sometimes go astray. For example, there's been a big emphasis on beet consumption in this group, but nobody mentioned the fact that beets are one of the highest vegetable with oxalates. So if you've got a oxalate calcium problem, you're going to have a great problem if you start eating beets, although you can ferment them and do some other things, but most beet solutions that are out there are not fermented. Okay, so that's my comment, but that's all of us participating in passing this around to our friends and colleagues. The other question is, it's never mentioned about translation. And are there any studies in translation which looks at the differences between males and females? I bet they're not, because all the studies in translation are not well funded. So that's my question.
You're talking about molecular translation, message to protein?
Yes.
Yeah.
The ribosome.
Yeah, well, I mean, I think we're making progress there, I think, because now there's proteomic stuff. And I agree with you, it's a problem. The correlation between message levels and protein levels is highly variable, highly uncertain. Using simply, RNA-seq to try to understand what's going on at the protein level has its problems. But I think we're getting better and better tools for looking at the actual proteome, and not just the transcriptome. So hopefully that will improve over time. I mean, there's an enormous study going on right now with about 10 institutions called the Longevity Consortium, and they're doing everything from genome sequencing species of exceptional gerontological interest to doing the proteome, the metabolome, all of the omics. I think ultimately we're going to learn something from that.
Are there any more questions? If not, we have about 5 minutes left. And I'll go ahead and ask Dr. Austed. I found the title and the content of the new book that you're writing pretty interesting. Maybe just with the last few minutes we have, give us a synopsis of sort of how you came to write that book and sort of what the general synopsis is of that.
Sure. So the book is called Methuselah's Zoo, and it's actually describes all of the animals that are of interest because of their exceptional longevity. So I basically marched through which animals are long-lived, which ones do things better than humans do at combating aging, how they do it, and what we might learn from each of these animals by doing it. So it's called Methuselah's Zoo because Methuselah from the Bible is the longest lived person that's ever been reported, 969 years. And it had a nice ring to it. So it's published by MIT Press. But it's just to warn you in advance, it's a book that's designed to be read by the lay public. It's not a technical book. That would be a different book, a book that actually MIT would like me to write the more technical version of. But I'm resisting that temptation because writing books takes a long time and takes time away from a lot of other things. However, I have to say it's not going to be out until August, but it's already being translated into Japanese, Korean, and Chinese.
I hope I'm allowed to say this. It also is available for pre-order on Amazon because I already checked. So.
Oh, is it? Oh, good. I'm delighted to hear that.
Yeah. So 3 or 4 minutes left. If nobody has any more questions, just a really fascinating lecture. Really appreciate your time, Dr. Austed. And then I'll go ahead and officially end this session if there are no more questions.
Well, thanks. It's been fun chatting with you. I wish we could have done it in person.
Yes, sir. Thank you.
All right. Goodbye.